is Felicia.
And my name is Paige, and we're here to introduce you to the amazing concept of mycoremediation as a component of oil spill reclamation.
The treatment of the Alaskan shoreline following the oil spill of Exxon Valdez in 1989 is a common example in which bioremediation methods got public attention. There are numerous other success stories of bioremediation in cleaning up chemical spills, including that of the 2007 San Francisco Bay Area oil spill. Mycoremediation is the application of fungi in the remediation of polluted terrestrial and aquatic environments. It involves the filtration of agricultural or industrial water runoff, the decomposition of hydrocarbon-based contaminants, and the removal of heavy metal from substrates by a fungal species.
One of the most potent chemicals in oil spills are called polycyclic aromatic hydrocarbons, or PAHs. Chlorotis austriatus, along with a number of other white rot fungi, are able to degrade persistent xenobiotic compounds, which are chemicals that humans encounter in the environment. This is due to the ability of their cell walls to excrete a mixture of enzymatic molecules. These enzymes include extracellular peroxidases, lignases, cellulases, pectinases, xanolases, and oxides. Of particular interest is the class of enzymes known as lignases, for their ability to degrade PAHs. Since both lignin and oil are PAHs, white rot fungi, low specificity enzymes are able to break down both. Furthermore, P. austriatus are shown to possess bioabsorption capabilities while in the presence of both cadmium and mercury. P. austriatus oxidative enzymes play a major role in the degradation process, along with organic acids and chelators, which are released by the mushroom. So we know that this works. The question isn't how it works anymore. It's about maximizing these observed effects through cultivation and application methods.
Additionally, fungi are opportunistic and respond quickly to environmental disturbances. These qualities, in conjunction with their ability to break down complex toxic molecules, is why the application and study of mycoremediation is so interesting. Mycoremediation of waste can be described by the processes of biodegradation and bioabsorption. Biodegradation describes the breakdown and recycling of complex molecules. This process produces much simpler non-toxic components such as carbon dioxide, water, and nitric oxide. Bioabsorption describes the process by which metals or pollutants are removed from the environment by mycelium. Due to the high uptake capacity and low cost of the raw material, bioabsorption has become a commonly used method of the removal of pollutants from the environment. Mycoremediation application technologies can be broadly classified as in situ or ex situ. Ex situ technologies involve the physical removal of the contaminated material for treatment process, and in contrast, in situ techniques involve treatment of the contaminated material in place.
In 2007, approximately 60,000 gallons of fuel was spilled along the shoreline of San Francisco. To combat this problem, mats made from human hair, yes, human hair, were used to soak up the spilled oil. These mats were then collected and layered with oyster mushrooms, or P. austriatus, and straw. These mushrooms were able to degrade the oil so successfully that after several weeks, the soil was good enough to be used for roadside landscaping. Quantitatively, a plot of soil contaminated with diesel oil and inoculated with mycelia from P. austriatus has been shown to decrease approximately 95% of the PAHs present over the span of a single month. Furthermore, over a 30-year period, Texaco discharged more than 18 billion gallons of oil waste into unlined pits in the Ecuadorian Amazon. Amazon MycoTalk Project, headed by Mia Maltz, is currently testing biofiltration systems which redirect contaminated water with oil through the fungi and then into bacteria and plants sequentially. Fungi play a pivotal role in the filtration systems by breaking down PAHs, such as petroleum, into bioavailable sugary food sources, which are then further degraded and transformed by bacteria and photosynthetic plants. After plants, microbes, and fungi are used to reduce the amount of petroleum in the environment using their design system, they are composted and added to the soil growing hardwood trees in order to continue nutrient cycling within the ecosystem.
The basic life cycle of fungi is to develop biomass capable of producing spores. Any fungi which is able to successfully do so in the presence of a contaminated substrate, soil, or fluid can be selected for most successful individuals. The colonizing mycelium can be subcultured and substrate contaminant levels stepwise increased in order to select for remediation and specific traits. It is by this basic method that nearly anyone with access to an initial culture can develop their own mycoremediation system. Oyster mushrooms, more specifically, are often propagated by mycelium on a substrate of waste product. Rice, straw, corn cobs, sawdust, or millet are shown to be very effective for cultivation, reliably inexpensive, and readily available.
Currently, incineration is the most common oil spill remediation practice used because of its effectiveness and ease of application. However, this method is extremely costly as it rarely truly remediates a zone, as it leaves a residual mess which later needs to be dealt with. It has become essential to discover an alternative remediation method which is both affordable, efficient, and has a significantly lower environmental impact. Paul Stamets, a renowned mycologist especially interested in mycoremediation, dreams of an establishment of mycological response teams as first responders to contaminated regions. Further enthusiasm is shown by American biologists associated with the Amazon MycoTalk Project. Statements such as mushrooms are the world's greatest recyclers as they eat trash, and the hope that one day people would be able to remediate their own land are specific examples. If you're not convinced by the amazing implications of mycoremediation, then let Paul Stamets convince you himself in his TED Talk, 6 Ways that Mushrooms Can Save the World. Google it.
Rice, straw, corn cobs, sawdust, or millet are shown to be very effective for cultivation. What is millet?
It's millet.
What?
Millet. If you don't succeed, try again.
Until you die, apparently. Okay.